I'd like to talk to you about our research project entitled Detection and Monitoring of Spinal Cord Injury using somatosensory evoked potentials. This research project is conducted at the American University of Sharhaj, and it is in collaboration with the Johns Hopkins College or School of Medicine in the United States. Spinal cord injury is one of those problems where of course it is detected using standard techniques in hospitals, primarily using imaging techniques, and there is still no accurate quantitative measure of the level of spinal cord injury. So what we're proposing in this project is to provide specialists and doctors with a quantity that would reflect the level of spinal cord injury following a major accident. So since we cannot conduct these experiments on humans, we have an animal model, which is basically rats. So we conduct our experiments on rats. And what we do, we use evoked potentials. So we stimulate the forelimbs of the animal, and the forelimbs and the hind limbs. And then we detect the evoked potential from the scalp of the animal. We take the signal to the computer after amplifying it with an EEG amplifier. The evoked potentials, of course, resulting from stimulating a normal pathway has a certain pattern, while the evoked potentials resulting from stimulating limbs with spinal cord injury have a different pattern. So since we do not have a reference for normal evoked potentials, usually after injury of humans, which is ultimately going to be the target of this project, we compare the forelimbs to the hind limbs using a technique, we call it a spectral coherence technique. The good thing about the spectral coherence that it provides the doctor with a quantity, a number, that varies between 0 and 1, where 0 means no correlation, no coherence between the forelimbs and the hind limbs, and 1 means 100% correlation. After spinal cord injury, especially if it's a severe spinal cord injury, we usually end up with a very small amplitude signals relative to a big signal that's usually obtained from a normal normal stimulation. This picture also shows the signals detected from left and right forelimbs and using different degrees of spinal cord injury. Now, the spectral coherence is displayed as a function of time. Here we take the spectral coherence of evoked potentials measured before injury, because we conduct experiments on animals under controlled environment. We have spectral coherence before injury, like a few days after injury, 1 week after injury, several weeks after injury. So now, for the case when there is a mild injury, just a very mild injury, we measure that by the weight of the that is dropped on the exposed spine of the animal. Before injury, you see spectral coherence that is relatively high, which is close to 1. But immediately after injury, a few days after injury, you see a drop down to around 0.4. Now, in case of a mild injury, we see a recovery of the coherence that may reach up to 0.9 here, or just above 0.8, which reflects a normal conduction and normal function of the spinal cord. However, in case of a severe spinal cord injury, this number drops down to actually around 0.4 here, which reflects severe spinal cord injury. The good thing about this technique that it provides a non-invasive method for studying and quantifying the level of spinal cord injury, and it can be conducted by non-specialists like nurse, not necessarily a specialist, or any technician or nurse can actually conduct this experiment. Thank you very much for listening.